So our topic for today is nucleic acids, and you can see that word nucleic acids right here. Nucleic acids, of course, are DNA and RNA. DNA stands for deoxyribonucleic acid, RNA stands for ribonucleic acid. Both of them are big macromolecules essential for life in our bodies. Both of them are nucleic acids. We're also going to discuss today what nucleic acids are made of, and it's pretty simple. A nucleic acid is made of 5 carbon sugars, a phosphate group, and a nitrogenous base. Now this whole thing here, if I were to put a box around this whole thing would be called a nucleotide. So we're going to discuss that in detail in a little bit. Okay, so what's a nucleic acid? It's a large organic molecule that stores genetic information, like DNA. Your DNA stores genetic information. It also transmits genetic information. That would be your RNA. Your RNA transmits genetic information. The DNA stores it. Okay. Nucleic acids play a central role in building proteins and other biological structures. And so the take home from this, now here's the big picture. You ready? Here's the big picture. In order for us to be alive, right, our bodies have to make certain proteins. Our bodies are instructed, right, to make those proteins through our genetic code. The instructions to make those proteins are held in your DNA in every cell, right, in the nucleus of every cell. Now, the DNA just is the code, right? So it's like if you had to put a piece of furniture together, right, you'd get a box with all the pieces, and inside the box would be the instructions, right? So the DNA are like the instructions, but then the furniture is not going to put itself together. Someone has to come along and read the instructions and then do the work. So the DNA is the instruction booklet, and your RNA comes along and reads the instructions and then tells the body how to put the proteins together, each cell. Okay, so the DNA holds the instructions. The RNA is able to read the instructions, and then the RNA is able to tell the cells in the body how to build the proteins for us to stay alive. So that's the big picture of why we're doing this. So the question is, what are nucleotides made of? The answer is the question is, what are nucleic acids made of? And the answer is nucleotides. So that's a new word for you, nucleotides. And those are what the big nucleic acid is made of. So a big strand of DNA or a big strand of RNA is made up of a bunch of nucleotides. And you can see here I have a nucleotide circled, okay, and it encompasses, if I pick one that's not covered up, it encompasses this entire piece. So this is a nucleotide. Let me get my pen and I'll circle it for you. Right, this is a nucleotide. This is a nucleotide. Notice I'm circling the blue strip as well. That's a nucleotide. This is a nucleotide. This is a nucleotide. Same thing on the other side. Here's one. I'm going to skip each one so it's easier to circle them, but here's a nucleotide and here's a nucleotide. Now each one of these nucleotides is made up of 3 things. Made up of a phosphate group, a sugar, and this thing here, which we call a nitrogenous base. Okay, so each one of these red circled structures called a nucleotide is made up of 3 things: a 5 carbon sugar, a phosphate group, and a nitrogenous base. Here's the phosphate group in yellow, here's the sugar in purple, and here's the nitrogenous base. Okay, now the next thing I'm going to do is change the color. We're going to make a big old mess out of this. And let's use green. Okay, no, I don't want to use green because that would be the nitrogenous base. We'll use purple. Okay, so this blue strip here, right, that you can see that I'm circling, that blue strip right there is called the sugar phosphate backbone. So hopefully I can do this. Sugar phosphate, that's a P, sugar phosphate backbone. So that blue strip is the backbone of the DNA. It's made out of the sugar of one nucleotide and the phosphate of another nucleotide. Now, I'm going to change my ink color to green because the next thing we're going to do is I'm going to show you where the nitrogenous base is. So this part, the nitrogenous base part of the nucleotide, is right here. It's this kind of ribbon looking thing or strip coming out. So this thing that I'm circling in green, that's the nitrogenous base, and it's gray, right? And you can see it's inserted right there. You can see how it's inserted into the backbone. So this green part is called the nitrogenous base. Okay, so bottom line here, what I want you to take home from this, after I've made a huge mess out of this, is that each section, right, of the nucleic acid, so this whole thing is a nucleic acid, each circled section, right, including the gray part and the blue part, that's called a nucleotide. And you can see it's circled right here. This whole thing right here is a nucleotide. And each of these, each nucleotide, is made out of a 5-carbon sugar, a phosphate group, and a nitrogenous base. And of course, if you are the kind of person who likes to see all of that in writing, I put it right here. So you might want to take the time here to just read this carefully. Nucleic acids, I'm going to point at the picture while I read this bottom part, right? Nucleic acids are made up of, so nucleic acids are made up of smaller subunits called nucleotides. Okay, nucleic acids are made up of smaller subunits called nucleotides. Nucleotides are made up of a 5-carbon sugar, a phosphate group, and this thing here called a nitrogenous base. The sugar molecule of one nucleotide bonds with the phosphate molecule of another nucleotide to form the sugar phosphate backbone. You can see the sugar phosphate backbone here on the right in blue, and you can see the sugar phosphate backbone here on the left. If you could think of DNA as a ladder, these would be the rails of the ladder that you hold on to when you're climbing up the ladder. So that sugar phosphate backbone makes up the backbone of the DNA molecule. Okay, so I want to take you to, if I could get this to work on my screen here, I want to take you to lesson 1.02, page 3 of your course. And what you should see, right, if I go to class home, and then I click on today's plan, and I click on today's lesson, which is 1.02. If I click on the Explore section, and then I go to page 3, I want to show you this link right here, the structure of DNA. So this is what DNA looks like, and it says down here at the bottom, DNA's unique structure is formed by linking large numbers of individual nucleotides. When the 2 nucleotide strands of a DNA molecule are untwisted, they resemble a ladder in shape. And if you pull it apart, a DNA molecule consists of 2 strands of individual nucleotides. You can see the nucleotides circled here, the gray part and the blue part, linked together and twisted to form a double helix. So each one of these is an individual nucleotide. Notice the blue part is circled as well. The blue section, that's the sugar phosphate backbone of the DNA molecule, and then these gray rungs are the nitrogenous bases. So this is the sugar phosphate backbone, and each one of these gray rods, or it looks like a ribbon maybe, are the nitrogenous bases. Put together, the whole thing, right, the backbone part and the gray part, is called a nucleotide. When you take a whole bunch of nucleotides and you put them together into a large strand, and you stick them together, and then you twist them, That's what a DNA molecule looks like. All right, so here's DNA again. It's a nice, beautiful picture, I think, of DNA. Of course, DNA actually isn't this color when we, you know, when we're looking at it in our bodies. But what I want to show you here, and I want to point out, is if I use my little laser here, this red ribbony looking thing is one side of the DNA molecule. That would be the backbone, sugar phosphate backbone. And then we have the blue side, which is the other side. That's the other rail of the ladder, if you will, the sugar phosphate backbone. So I know we say that DNA has a sugar phosphate backbone. There's actually like two sides to that backbone, right? Each side of the ladder. And then the rungs coming across the ladder, notice they're different colors. This one's pink or magenta, and it's attached to a blue one, and then this yellow one is attached to a green one. And if you notice in this picture, they're always attached to the same color. So pink is always bonded to blue, pink to blue, pink to blue, or actually pink to blue, pink to blue, pink to blue, right? And then the yellow one is always bonded to a green one. So there's four colors. There's pink, blue, yellow, and green. And those four colors represent the four different nitrogenous bases that hold the DNA molecule together. And now the color is different in every time someone draws a DNA molecule, they may pick a different color. So instead of using colors to represent the nitrogenous bases, we're actually going to give them names. And I'm going to show you here on this next slide what they're called, and then we're going to go back and label them. Okay, so what you see here is there are four nitrogenous bases. Those are those gray rods coming across, right? They could be adenine, cytosine, guanine, or thymine. Now what I will tell you is that, I better change my color because you won't see it if I pick that color. What I will tell you is that adenine always bonds to thymine, and cytosine always bonds to guanine. So for example, if this one were adenine, change my color again. If this were adenine, so I'm going to write an A for adenine, this nucleotide over here, whoops, would always be thymine. If this one were cytosine, this one would always be guanine. Okay, again, adenine always bonds to thymine, cytosine always bonds to guanine. A to T, C to G. That's how I remember it. A to T, C to G. Okay, so let's go back one slide, and we're going to go ahead and we're going to link these up here. Change my color. Okay, so if this one is always adenine, this one would always be thymine. So if the pink ones are always adenine, the blue ones are always thymine, right? A always bonds to T, and C always bonds to G. Now that's not a G because it's green. It just happened to work out that way. Okay, so all my G's would always bond to C's, and all my T's over here on the right would always bond to A's. That's how DNA gets put together. Remember, A to T, C to G. A always bonds to T, A always bonds to T, C always bonds to G. And you can see it here on the right. We have a sugar phosphate backbone, the sugar phosphate backbone, and we have a nitrogenous base of cytosine bonding to the nitrogenous base of guanine. Thymine always bonds to adenine. Guanine and cytosine always bond together. Adenine and thymine always bond together. Again, sugar phosphate backbone, nitrogenous base bonded to another nitrogenous base. And if we really want to dig deep here and label this, I'm going to put hopefully a square around this. This part here, whoops, is one nucleotide, And then over here would be another nucleotide. They're not supposed to overlap, but I'm having a hard time using my pen, right? One nucleotide, two nucleotides. One nucleotide, two nucleotides. Notice I included the sugar phosphate backbone and the nitrogenous base within the nucleotide. So the nucleotide is the whole thing. It's the backbone plus the nitrogenous base right there. All right, the last thing I want to show you before we, see if I can move that over. There we go. Before we move on to the next part of our lesson today is I wanted to give you a little history of the discovery of DNA, and it started in 1949. Erwin Chargaff discovered that the number of adenine bases always equals the number of thymine, and that cytosine always equals the number of guanine. And that's how he figured out that they paired up with each other. So if there were like 500 adenines, there'd also be 500 thymines. And if there were like 200 cytosines, there'd also be 200 guanines. So that's how he figured, well, if there's 500 adenines and 500 thymines, they must go together. And if there's like 6 cytosines and 6 guanines, they also must go together. So that was the first part of discovering DNA. And then in the 1950s, Rosalind Franklin was the first person to take an X-ray photo of DNA, which gave us a hint to that double helix shape. And then in 1953, Watson and Crick were able to determine, they were the first to actually build a model, and here it is. They were the first to actually build a model of DNA in that double helix shape. So if you're wondering who these guys are down here, there's a show that used to be on just a couple years ago, not too long ago, called Miles from Tomorrowland, and they actually named their commanders. This is like a two-headed alien, and they're named Watson and Crick after the first two folks to develop a model of DNA.